Thank you, Sydney, and thank you to everyone for joining us today. Over the next 15 minutes, we are going to discuss a monumental paradigm shift currently underway in neurodegenerative disease research, the transition toward accessible, scalable, blood-based biomarkers for Alzheimer's disease. To understand the magnitude of this shift, we first have to confront the reality of the global diagnostic gap. Right now, Alzheimer's disease remains profoundly underdiagnosed, with less than 25% of individuals with dementia ever receiving a formal diagnosis. Even more alarming is the fact that more than 90% of people with mild cognitive impairment, or MCI, a condition that often progresses to Alzheimer's, never receive biological testing. Historically, we have relied heavily on amyloid PET imaging and cerebrospinal fluid, or CSF, testing. While these are the gold standards, they come with immense barriers. PET scans are expensive, require specialized equipment, and are severely limited geographically. CSF testing, on the other hand, is inherently invasive, leading to high patient hesitancy. Because of these limitations, rural, minority, and low-resource populations have faced disproportionate diagnostic barriers. This is where blood-based biomarkers fundamentally change the equation. A simple blood draw is non-invasive, cost-effective, and can be performed in virtually any clinical setting worldwide. Today, two phosphorylated tau biomarkers, p-tau217 and p-tau181, have emerged as the most widely validated plasma markers for detecting Alzheimer's disease pathology. They allow clinicians to screen patients earlier, streamlining referral pathways and closing the equity gap. In fact, we now have FDA-cleared assays anchoring a modern diagnostic workflow. Roche's Elxis p-tau181 serves as an early rollout test in primary care, while Fujirebio's Lumipulse p-tau217 A-beta42 ratio provides highly specific rule-in confirmation for specialty settings. P-tau217 achieves AUC values up to 0.94 to 0.98 for distinguishing Alzheimer's disease from non-AD disorders. Together, these biomarkers support a tiered diagnostic workflow from rule out in primary care to rule in specialty settings. However, translating these promising assays into universal clinical tools requires a robust scientific foundation. Today, I will outline the three critical pillars necessary to validate these, as well as any diagnostics: pre-analytical rigor, the availability of longitudinal specimens, and the depth of clinical and demographic data. These pillars are not just scientific requirements, they are prerequisites for regulatory validation and clinical adoption. Let's move to our first pillar, the quality of the starting raw material and the importance of pre-analytical rigor. When we look at the biology, p-tau217 and p-tau181 we've left different points along the Alzheimer's cascade. P-tau217 is highly specific to the AD type tau pathology and correlates strongly with amyloid and tau PET imaging, elevating years before symptoms appear. In contrast, p-tau181 is a sensitive marker of Alzheimer's-related tau change and is associated with disease progression. But to detect these markers accurately in blood, we must account for their unique stability profiles. P-tau181 is notably robust. It maintains stability at room temperature, tolerates multiple freeze-thaw cycles, and may show good agreement across metrics on some assay platforms, though plasma remains the preferred matrix in many workflows. P-tau217, however, requires much stricter handling. It performs best when measured in ETA plasma and is quite sensitive to delayed centrifugation, particularly when assays incorporate A-beta42 ratios. Despite these nuances, p-tau217 demonstrates excellent long-term stability when stored at -80 degrees, which is critical for reliable biobanking. If these pre-analytical variables are not controlled, data quality can become unreliable and difficult to interpret across assays and study sites. Standardized collection and processing workflows are absolute prerequisites for ensuring cross-assay reproducibility and establishing clinical reliable cutoffs. This means strict control over tube type, centrifugation timing, freeze and thaw history, and time to spin requirements. Furthermore, high-quality raw material is essential for assay development itself. Developers need rigorously controlled materials like high, mid, and low p-tau plasma pools, as well as matched CSF and plasma quality control ladder sets. These are the foundational tools required for reproducibility assessments, linearity, and precision studies, and ultimately regulatory submissions. While I described specifics for p-tau, these needs are consistent for any diagnostic development. This brings us to our second pillar, the vital need for longitudinal specimens. Alzheimer's disease is not a static condition. It is a progressive continuum. A cross-sectional snapshot, a single blood draw at one moment in time, misses the dynamic trajectory of neurodegeneration. One of the greatest advantages of blood testing over invasive CSF or expensive PET scans is that blood samples can be collected repeatedly over time, enabling true longitudinal monitoring. To truly understand biomarker trajectories across preclinical stages, MCI, and false Alzheimer's, researchers need access to multiple time point collections. At BioIVT, for example, longitudinal sampling is conducted at 6, 12, 18, 24, 30, and 36 months, with some donor cohorts followed for up to 6 to 8 years. This extended time frame allows for detailed analysis of early biomarker changes, therapeutic response patterns to new anti-amyloid drugs, and long-term disease progression. This is particularly critical for evaluating therapeutic response in anti-amyloid and emerging anti-tau therapies. Again, while describing specific needs for Alzheimer's disease, these needs translate to any diagnostic related to long-term patient care and health, such as oncology, autoimmune, and metabolic, for example. But longitudinal blood samples alone aren't enough. We need matched sets. By providing matched plasma, serum, CSF, and PBMC collections, we allow researchers to cross-validate experimental blood biomarkers against gold standard CSF measures, such as the A-beta-42/40 ratio, p-tau 217, p-tau 181, total tau, and neurofilament light chain. These sets allow cross-validation across gold standard CSF biomarkers and support cross-platform comparability and harmonization efforts. This is especially important when comparing performance across platforms like Simoa, Lumipulse, and Alzheimer's. These matched sets are exactly what the field requires to harmonize assays across these different technical platforms. Finally, and we hinted at this in the previous slide, we reach our third pillar, the extent of clinical and socio-demographic data. If we want blood-based biomarkers to bridge the global detection gap, we must ensure these tests work accurately across all populations. Currently, there are major open questions regarding how biological variables such as race, specific comorbidities, and socioeconomic factors might influence clinical biomarker cutoffs. To resolve this, recruitment must actively prioritize underrepresented groups, including African American and Hispanic donors, rural populations, and communities historically marginalized by diagnostic gaps. Diverse donor representation is non-negotiable for establishing universally applicable thresholds of diagnostics. Additionally, specimens must be paired with deep clinical and cognitive metadata, such as BioIVT's cognitive rating scale data. This allows for stratified donor selection. Investigators need the ability to select specific biological profiles for their research, For example, requesting donors who are p-tau217 positive with low A-beta42, or perhaps dual negative controls with unimpaired cognition. This enables stratified cohort selection for early detection, differential diagnosis, and assay validation studies. Looking ahead, we are moving beyond just p-tau. The future of precision medicine diagnostics relies on comprehensive ATN profiling, combining amyloid, tau, and neurodegenerative markers. This means integrated p-tau with markers like gliofibrillary acidic acid, or GFAP, for astrocyte reactivity, and neurofilament for axonal injury, alongside broader proteomics and genomics. This aligns with the NIA-AA ATN framework, which is increasingly guiding both clinical trials and diagnostic strategies. Developed by the National Institutes of Aging, or NIA, and the Alzheimer's Association, AA, in 2018, it shifts the definition of Alzheimer's disease from a clinical syndrome to a biological construct. This framework defines Alzheimer's disease biologically using biomarkers rather than clinical symptoms, categorizing individuals by amyloid, tau, and neurodegeneration status. By combining these three categories, positive or negative, researchers can then identify 8 different biomarker profiles from normal to early pathologic Alzheimer's disease changes to Alzheimer's disease to non-Alzheimer's disease related changes. And as before, while this is specific for Alzheimer's, the same types of multi-omic contexts are being developed around many diagnostic workflows. In conclusion, the rapid evolution of p-tau217 and p-tau181 is fundamentally shifting the landscape. But to finalize universal cutoffs and harmonize assays, the scientific community relies on a robust foundation. By ensuring pre-analytical consistency, leveraging longitudinal matched biospecimens, and demanding diverse participant representation, we can validate diagnostics that are not only accurate, but globally equitable. Organizations like BioIVT are providing this biological and operational infrastructure, enabling the advancement of the next decade of Alzheimer's research to enable not just scientific validation, but regulatory alignment and global clinical adoption. Thank you for your time today, and for more information on BioIVT's Alzheimer's and other neurological disorder biorepository and sample availability, please visit info.bioivt.com/CFNE.